A recently developed tool enhances and streamlines the process of CRISPR genome editing, resulting in increased speed and optimization. Greetings everyone. Today we will be delving into an exciting topic. The focus of our video centers around the groundbreaking work of researchers at the Center for Advanced Bioenergy and Bioproducts Innovation, CABI. They have recently developed a remarkable tool known as CRISPR-COPIES. This tool has the incredible ability to pinpoint suitable chromosomal integration sites for genetic engineering in any organism. In a matter of just 2 to 3 minutes, CRISPR-COPIES can identify neutral integration sites across the entire genome of most bacterial and fungal genomes. This eliminates the need for labor-intensive manual design of components for CRISPR/Cas-mediated DNA integration. The implications of this tool are vast, as it can be utilized to enhance biomass yields, bolster pest resistance, and fortify environmental resilience in crop engineering. Additionally, it can be employed to engineer cells that exhibit significantly higher yields in the conversion of biomass into valuable chemicals. So, without further delay, let us dive into our discussion. According to Huimin Zhao, the leader of CABI's conversion theme and the Stephen L. Miller Chair of Chemical and Biomolecular Engineering, CHBE, at the University of Illinois, the tool known as CRISPR-COPIES has the ability to rapidly pinpoint suitable locations on chromosomes for genetic manipulation in any organism. This innovative tool will greatly expedite our efforts in the field of metabolic engineering, especially when it comes to modifying non-model yeasts for the efficient and economical synthesis of chemicals and biofuels. The field of genetic engineering has been completely transformed by the advent of gene editing, providing scientists with unprecedented abilities to comprehend and manipulate genetic data. Through this innovative method of genetic modification, researchers are able to incorporate novel characteristics into organisms, ranging from enhanced pest resistance to the production of valuable biochemical compounds. The ability to perform accurate and specific genetic modifications is made possible by CRISPR-Cas systems. Nevertheless, the challenge of identifying the most suitable locations in the genome for these modifications remains a significant and unresolved issue. In the past, researchers would manually search for potential integration sites and then evaluate their viability by integrating a reporter gene to measure cellular fitness and gene expression levels. This method was both laborious and demanding in terms of time and resources. In order to tackle this particular obstacle, the CABI team has devised CRISPR-COPIES, a computational pipeline known as Computational Pipeline for the Identification of CRISPR/Cas-facilitated integration sites. With an impressive turnaround time of just 2 to 3 minutes, this innovative tool is capable of pinpointing neutral integration sites across the entire genome for the majority of bacterial and fungal genomes. Ashutosh Boob, a PhD student in CHBE at the University of Illinois, and the main author of the study, compared the arduous task of manually finding the integration site in the genome to searching for a needle in a haystack. However, with the implementation of CRISPR-COPIES, this challenge is transformed into a navigable realm, allowing researchers to easily locate all the needles that meet their specific requirements. The researchers showcased the adaptability and expandability of CRISPR-COPIES in a study published in Nucleic Acids Research. They examined integration sites in 3 distinct species, namely Cupriavidus necator, Saccharomyces cerevisiae, and HEC293 T cells, to demonstrate the versatility of CRISPR-COPIES. By utilizing the integration sites identified through CRISPR-COPIES, they successfully engineered cells that exhibited enhanced production of 5-aminolevulinic acid. This biochemical compound holds significant value in both the agricultural and food industries. Furthermore, the CRISPR-COPIES team has developed a web interface that is designed to be user-friendly. This highly accessible tool allows researchers to utilize the application, even if they do not possess extensive knowledge in the field of bioinformatics. CABI's main goal is to utilize non-model yeasts for the production of chemicals and fuels derived from plant biomass. However, scaling up the production of biofuels and bioproducts from cost-effective feedstocks poses a challenge due to the limitations of genetic tools and traditional genome editing techniques. CRISPR-COPIES offers a solution by allowing researchers to efficiently identify specific genomic locations for targeted gene integration. This streamlined process enables the identification of stable integration sites throughout the genome, while also eliminating the need for manual component design in CRISPR/Cas-mediated DNA integration. The application of this tool in crop engineering allows for the enhancement of biomass yields, pest resistance, and/or environmental resilience. Similarly, when it comes to the conversion of biomass into valuable chemicals, such as through the utilization of the yeast S. cerevisiae, CRISPR-Cas9 can be employed to engineer cells that yield substantially higher quantities. The primary objective of this adaptable software is to streamline and expedite the process of strain construction, resulting in significant time and resource savings for researchers. Its wide-ranging applicability extends to researchers in various fields, including academia and industry, who can leverage its capabilities for strain engineering in biochemical production and the advancement of transgenic crops. Contributors to this study consist of THBE PhD student Jixin Zhu, visiting THBE student Patarawan Intazian, and bioengineering PhD student Guanhua Shun. Additionally, the team includes software developers Manan Jain and Vasily Petrov from the Carl R. Woese Institute for Genomic Biology, IGB, Biofoundry manager Stefan Lane from IGB, and CABI postdoc Shi Itan. To support our channel's growth and ensure broader awareness, kindly hit the like and subscribe buttons. This will help us reach more individuals and disseminate valuable information. Thank you in advance.